Hello, my students. Once again, welcome back. So this is the uh, second video of this particular series. Now, in this video, we will be talking about the lactose operon. That means the gene regulatory system, which is visible in a prokaryotes, mostly the bacteria. So before we begin with the whole lot of the structural details, functional details, and all stuff. First, let's understand the meaning of Lac Operon. Now, when we talk about operon means it's a unit of regulation. Now, when we talk about the word regulation, it actually means the gene expression will be regulated. Again, the same thing, correct time, correct amount, correct version. Everything will be regulated. So, this is the regulation. So, the unit of the regulation is known as the operon. This is first thing. Second, the word lax stands for lactose. Now, as you know, lactose is a type of carbohydrate. So, when we think about the carbohydrate, the first thing that comes in my mind is it acts as an energy currency of the cell, right? It will give you the energy to do a lot of activities for your survival. So, lactose typically is a disaccharide which will be broken down into some monosaccharide known as glucose and galactose, which can be further utilized for the respiration purposes so that you can liberate the energy which is trapped inside those monosaccharide. This is something very basic. We have also seen in the previous video that it's an example of polycystonic gene. That means a single promoter and more than one structural gene. Right? So, in due course of time, we will see that how it supports this particular word polycystone. Now, to begin with the example of lactose operon, let me tell you one thing in the very beginning, that it's basically the utilization of lactose by a cell. Why the cell will be utilizing the lactose? Of course, for the sake of energy. Second thing, we will see in due course of time, if at all there is a presence of lactose, the whole system, the whole machinery will be switched on. So, if there is lactose in the medium, this operon will be switched on. If there is no lactose in the medium, it will remain switched off. That means we can take this example as our inducible regulatory system. That means if you want to switch on an operon system, you have to give some product. So it's a kind of inducing something. So it's an example of inducible operon system or the inducible regulatory system. So once again, if you have the lactose, it will be switched on. If you do not have the lactose in the medium, it will remain switched off. Second thing, so we have been talking about the operon, the unit of the regulation. How does it look like? Is it like if you take any part of the DNA, and can I call that particular part of the unit? No. That means some criteria is there. Now, what's the criteria? Criteria number one. First, there will be the presence of some structural genes. If we are talking about the regulation in case of prokaryotes, it will be a number of structural genes, because that is how you get the concept of polycystonic. If you are taking this example for the eukaryotes, it will be a structural gene. But moral of the story, structural gene has to be there. So criteria number one, I have got the structural genes. As far as Lac Operon is concerned, I have got three structural genes, and they are Lac Z, Lac Y, and Lac A. Now, why structural? That means each one of them, they have some specific function to perform. So if you talk about lac Z, the Z structural gene, it is responsible for the production of an enzyme known as beta galactosidase. What's the function? We'll talk about it later. Then you have got lac Y, which is responsible for the production of permease. It's a transport port, I should say it's a channel protein. Then you have lac A. This is responsible for the production of transacetylase. That means 3 structural genes, 3 different products and specific. So they are said to be the structural gene. Now upstream to that, in the same segment of the DNA, upstream to that, you have 2 more regions. One is the operator region. I've written in the like the abbreviation, like O stands for operator and all stuff. So operator region is responsible for how much protein will be produced. Or typically, this particular region is responsible for the regulation of a transcript. How much mRNA will be produced? So, how much protein will be produced? That is actually taken care by the operator region. And somewhere down the line, you will see, like this operator region is a kind of overlap with the promoter region. Now, what's the promoter region? Promoter region is further upstream to the operator region. This promoter region is a region in the DNA segment where a particular enzyme will come and bind. So in the previous video, we have talked about formation of the mRNA from the DNA via transcription. So if you want to produce an mRNA from this, you need to have a typical enzyme known as polymerase, right? So this is an area, at the area of P, in the promoter region, the polymerase enzyme will come and bind, right? Further upstream to that, you have 2 more regions. One, there is one more structural gene that is known as lac I. Now I stands for inhibitor, or you can say repressor. That means somewhere down the line, this particular gene is responsible for the production of some inhibitory protein. That means it will inhibit something. And upstream to that, you have the proper promoter for that. So you can see this is an example of monocistronic. Why? Because one structural gene, one promoter. This is an example of polycistronic. Why? More than one structural gene, one promoter. Right? So that's about the whole structure, I should say typical structure of a Lac Operon. Right? Now let's understand the function. See guys, what happens basically. So these are the lactose. Now first to think about, when does a cell require lactose? Well, in order to produce energy. Why does it require energy? For its survival, of course. Now if there is a condition where the cell is not having glucose, which is said to be the best chosen option for a cell, and suppose the cell is having lactose in the medium. So the cell wants to degrade the lactose into its subsequent form in order to release the energy. So for that to happen, I need to perform some task. And through that task, I'll make sure that I will degrade the lactose which is present in my body, in my cell, number 1. Number 2, I will make sure that I can uptake maximum amount of lactose. Why maximum? Maximum energy. So these 2 particular work will be taken care by the structural gene that I have. So again, what are all the functions I take care of? Number 1, lactose degradation. Why lactose degradation is important? If you degrade the lactose, you will be able to release the energy out of it from the glucose and the galactose, right? Number 2, maximum uptake of lactose from the surrounding medium. Right? Now understand. If there is lactose, then only it will make sense. Right? If there is no lactose, then of course unnecessarily will not perform all this task. So what the cell does, cell has developed the system in such a way that if there is no lactose, the whole mechanism will shut. It will not work. That means there won't be any lactose degradation because there is no lactose at all. Second, there won't be any uptake of the lactose. Again, there is no lactose at all. Now, how this will be accomplished? Here, the LAC-I gene, the inhibitory gene, comes into play. They are responsible for the production of one protein, which is inhibitory in nature. Now, this LAC-I inhibitor or inhibitory protein is allosteric in nature. It is said to be your allosteric in nature. What does that mean? It means that this particular protein will have more than one active site. It has something else other than active site. So, as you can see, this particular inhibitory protein has got two sites. One site is responsible, with the help of which it can get attached to the promoter site, as you can see I've written also. And the other side is responsible for the attachment with the lactose. So it's a kind of allosteric modification, or you can say an allosteric nature of that particular protein. Now what happens? If there is no lactose at all, that time this inhibitory protein will go and it will get bind itself with the help of this site, promoter binding site, in the promoter region. So what will happen? If the promoter region is blocked, so this polymerase enzyme won't be able to come here. Now it can't sit here. Why it can't sit? Because the promoter region is already blocked by this particular proteins, which is said to be the inhibitory protein. So why it has blocked? The reason is, if you still keep it open, it will come, it will bind, it will transcribe the whole DNA, the structural gene, and unnecessarily you will keep on producing the products. Again, wastage of energy. So to have a control over there, the cell has come up with a system in which no lactose, fine, let's shut down the system. If there is no laborer, if there is no labor in the factory, obviously it becomes meaningless to keep the factory open. So the factory will no more be functioning without the labor. Same thing is happening over here. Right? So again, no lactose, it will remain switched off. How? By this. So in the promoter region, the promoter which is responsible for the transcription of the structural gene, this will be occupied by the inhibitory protein. And this inhibitory protein will not allow the polymerase enzyme to come and sit. Typically it's an RNA polymerase, because at the end of the day it will be producing the mRNA transcript. Right? Now, suppose in the cell, inside the cell, a few lactose have come out, or they have synthesized, or from some other sources it has come. So one of the lactose molecule that I have drawn over here, it will come and it will attach itself to this particular site. So this, because this is the site which is responsible for the lactose attachment, because it's allosteric in nature. If this happens, the moment lactose gets attached to this protein, there will be a conformational change in this inhibitory protein. I repeat, there will be a conformational change in this inhibitory protein. So what will happen? The moment this protein undergoes a conformational change, it will fall off. It will get detached because this side now will get modified. Which side? Excuse me. This side will get modified. The side which is responsible for the attachment at the promoter level. And now, if this side has got changed conformationally, so what will happen? It will no more be in a position to get attached itself. As a result of which, this promoter, or I should say, this inhibitory protein will fall off. So now there won't be any inhibitory protein. That means what? The promoter region is free to catch the polymerase. So now this polymerase can come and attach here. The moment it comes and attach here, it will undergo the transcription. If the transcription takes place, you can produce the mRNA transcript. Right? From the mRNA transcript, you can produce these three different types of proteins. Number one, the Lac Z will produce beta galactosidase, which is an enzyme. And as you know, enzymes are proteinaceous in nature. This enzyme is responsible for the degradation of the lactose. I've told you the reason why. Lac Y is responsible for the production of another enzyme. Permease, it's basically channel protein. So through this protein, the cell can increase the uptake of lactose from the medium into the body. I should say into the cell, inside the cell. Transacetylase, in combination with the permease, it will perform the same thing. I can say it can, kind of accelerate the uptake of lactose from the medium inside the host cell, the cell of the consideration here. Now, the thing is, when it is happening, if there is lactose, so what happens? If there is lactose, the operon will be switched on. What do you mean by that? It means that the promoter region will no more be blocked by the inhibitory protein. Well, how that's possible? Because lactose will get attached itself at the lactose binding site, because it's allosteric in nature, I've told you already, and that attachment of lactose will bring some conformational changes, right? As a result of which, it will get detached, they will remain free, it will become free. The polymerase will come and it will get attached itself. Transcription will take place, formation of mRNA, and from the mRNA, right after the translation, you will have all these end products: the beta galactosidase, the permease, and the transacetylase. So, my students, you see one thing here. In this particular case, this operon can only become functional if you have lactose into the system. So if there is a lactose, that means if there is that condition, then only it will work, or else it will remain shut down. So it's an example of inducible system. So lactose will induce the whole mechanism to be switched on. Right? So that's about the Lac Operon. I hope you have understood the whole mechanism. Well, if you have any doubts, any questions, you can write all the questions down below in the section. And in the next videos, we'll be coming up with the regulatory system of the gene expression in case of a eukaryotic cell. Again, the same thing. Those who are watching my videos for the very first time, don't forget to subscribe to my channel. Share the videos with your friends, with your buddies. Like, dislike also if you don't like the videos, and let me know in the comment section below why you have not liked my videos. I should say, don't hesitate to come up with all the questions. Feel free to shoot me any kind of question. Till then, bye bye and take care.